Good morning, everybody. My name is Fizza, and I'm here on behalf of Professor Caetano-Anollés from the Evolutionary Bioinformatics Lab at the University of Illinois. So we'll be talking about our activities on blue waters. This is basically a summary of the past two allocations that we had on blue waters, including our current one. In order to study function of proteins, we cannot just look at protein structures. We have to account for the dynamics of the protein. So this is because proteins are not static entities. They are rather a dynamic ensemble of conformations in the cell. And it has been observed that these conformations are not just random, but rather there are some constraints acting upon it, evolutionary drivers, if you may. So this has been demonstrated by, I'll be showcasing some of the studies. There have been similar or common fluctuations in the dynamics of the Rossmann-like fold in proteins that's been observed by these two groups. And then there's this common sharing of dynamics among the members of the same protein family and the protein superfamilies when it was compared with unrelated and distant proteins. And also, there are these common dynamic properties that can be detected in proteins that have different structure but have a common function, as is evident from this diagram, that although both of these proteins differ in the structure, their dynamics are in the same space. So based on this, now since we specialize in evolutionary bioinformatics, we're interested in studying how how protein function is acting in evolution. So my colleagues published this study last year that used loops and domains of proteins in order to trace these evolutionary origins. Now, this is a bipartite network of protein loops, and now these protein loops are elementary functional loops. Now, the concept of elementary functional loops is that protein domains are basically collections of these canalized loop structures that endow function upon the protein. So my colleagues mapped this bipartite network with the domains that signify the evolutionary ages, so in order to see how function is evolving. So based on this study, they concluded that function is evolving in a highly modular fashion. Now, what this space lacked was the details of dynamics, and we would like to incorporate that details. So in order to do so, we applied for allocations on the blue waters, and so this is a summary of the two allocations that we received with regard to these experiments. So in total, we have performed 203 molecular dynamic simulations. In the first cycle, we performed, we looked at 87 loops and the simulations were 10 nanoseconds each. And in the second cycle, we looked at loops that were associated with the metaconsensus enzymes, and they were 116 in number. And these spanned about 50 to 70 nanoseconds each. So in the first cycle, we looked at amino-acyl tRNA synthetases. Now, this data, I had borrowed this data from the work of our colleagues that was published in 2013. So the conclusion of this study was that the amino-acyl tRNA synthesis are the best candidates to study the origin of the genetic code, and flexibility is the main driver of the evolution of the genetic code. And this flexibility is, of course, found in the protein loops. So the next data set was loops that were related to metaconsensus enzymes. So meta-consensus enzymes are a group of 6 enzyme classification enzymes that have been derived from a consensus of 3 established comparative bioinformatics methods that are based on sequence, structure, and metabolic reactions. So, this is by the group of Goldman. They hypothesized that these meta-consensus enzymes represent the repertoire of metabolic reactions in the most ancestral forms of life. So another point to note is that in the second cycle, we exclusively looked at single domain proteins because we believe that in the first cycle, when we did a quick global analysis, there might be some kind of noise that might be interfering with our values of variables. So based on that, so we wanted a means of quantifying and quantifying variables derived from those dynamics. So we borrowed the concept of Grubb-Muller and colleagues about the construction of the dynasome, that is a creation of a dynamic space. So they created this dynamic space in order to trace the dynamic fingerprints of proteins. And what they observed was that functionally similar proteins tend to cluster together, so which makes dynamics the best candidate, with not only just structure, to trace the evolutionary origins. And dynamic space is continuous. We'll revisit that in a bit. And that when they just consider the dynamic space, that is shown in C over here, that they could see clusters of proteins that had same function, but so which performed so they made predictions based on that, and this so their method performed slightly had a reasonable performance rate compared to the size of their limited data set. But when they combined the structure and the dynamics information, the prediction of function yielded better results than if you use either of the two alone. So based on this, we constructed our own modified dynasome. And the variables that we have used in this dynasome are the ND values, that is the evolutionary ages, and then mean RMSD, mean radius of gyration, and then the eigenvalues of the top 5 principal components. Now this was, they were derived from a PCA analysis on the motions of those proteins, that is the trajectories. And then the next variables are based on the network metrics. These networks were constructed based on the community dynamics of those protein loops, based on the trajectories. And so we looked at the network diameters, average path length of those networks, and then clustering coefficient and how modular they were. And then we also did power law fitness tests, because we wanted to see if there was the presence of scale-free properties. And also, we were looking at average path lengths and clustering coefficients for whether there was some small world properties present. Now, we also wanted to see whether these networks were, maybe they were behaving in a random manner. Maybe these dynamics were, the community structures were just randomly forming. So for that, we performed the Bartel's rank test, from which we derived the statistic. And then in the presentation last year, we noted that there was this bias of betweenness values, that there was these loops with a certain function that had higher betweenness values at the N termini compared to the C termini. So in order to look at that, we split the betweenness values of the network into regions based on the residues where they're located, whether they're in the N termini, N or C terminus, or they're in the loop region. Same goes for measuring closeness and degree centrality. Now, these are all normalized measures. So a quick recap of what we had done last year. So last year we had performed the individual analysis for all these loop structures. Now this analysis is based on the very first allocation that we had gotten, that is with reference to the amino acid tRNA synthetase loops. So this is the community structures I was referring to. Now these are communities formed on the basis of the dynamic cross-correlations of these residues, such that this shows that whether the residues are moving together or moving in an anti-correlated manner during the trajectory. And this is the PC analysis that we performed, from which we derived the eigenvalues. And so this is based on the motion of the protein residues during the trajectory. To give an overview, so we had this is the breakdown of how the gene ontology functional annotations were represented in our set. Now, this is something to note that we have 87 loops, but some of the loops may have multiple functions assigned to them. So this is why the number is a bit higher. So as you can see, while 4 out of the 6 categories are not that well represented, we will mostly try to focus on the 2 categories that are well represented in our preliminary analysis. Okay, so as I mentioned about the network metrics, since binding and catalytic activity are somewhat well represented, we did notice a bias that the binding values, the binding C-terminus between these values are slightly higher than the N-terminus values compared to the catalytic activity, which has slightly higher values for the N-terminus and slightly lower values for the C-terminus. So this is something you would like to look at and perhaps make global statements if we have a more representative data set. Another thing to note is that this is somewhat expected, that since the loop regions are the most highly flexible parts of the structure that we are simulating, the Dern region, that is, so they have the highest betweenness and degree centrality values, and their spread is they're well distributed, or widely distributed, I would say. However, when you look at average closeness, it's steady for all 3 secondary structures types that we are looking at in the loops. And so now we needed some way of visualizing the design space of dynamics in the community for these community networks. So for that, we borrowed this concept from the Zukos naming rules. It's also known as the Zukos triangle. This is used for domain name addresses for naming systems. That at the edge of there are 3 properties attached to a name address. That it can be either memorable, that humans are easily able to remember it. It's decentralized. There's no single authority controlling the naming, and it is secure. That is, it is robust to attacks. So, what this Zukos triangle postulates is that it is impossible for a name to fulfill all 3 of these criteria. It can fulfill 2 at most. So, based on that, we interpreted the work of Solé and Wavardi in the context of complex networks, that they represented randomness, modularity, and heterogeneity in that 3-dimensional space, in a triangular space, to describe networks, how they're placed. So in this context, if randomness equates to security, because since it's random, it's hard to decipher the pattern and to infiltrate the system or the network. And then modularity signifies decentralization, that there's no central authority dictating that holds the power in a network. And scale-free networks have these popular nodes, and so that is why you're able to kind of remember them in that context, that they stand out much more. So based on that, we tried to plot to plot network metrics based on this three-dimensional design space based on the work of Anollés and Valverde. So in order to measure randomness, we used the Bartel's test statistic, and for modularity, we used maximum modularity scores to see how modular they were. And then for heterogeneity, we had, as I mentioned, we had performed power law fitness tests. For that, so on this axis, we have used the power law exponent. We have been plotting the power law exponent, so which is otherwise known as alpha. Some people call it gamma. So in order for it to be in the scale-free range, it needs to be from 2 to 3, although that is not true for most networks. But in our case, we were able to find two data points that were in that region. It's 3D, so it's unable to capture that. So based on this design space, we can see that there's a tendency towards being pushed towards modularity. That is, modularity equates to flexibility when we look at a biological context. So, which is expected because we are looking at loop regions that contain the most flexibility in a protein structure. So, since this is an ongoing work, this was just a preliminary analysis for one of the set of simulations that we performed, the first one. We will extend this analysis once it is complete. We will extend it to the second set of our simulations that are based on the meta-consensus enzymes. So what we're looking at right now, the next steps involve that we're thinking of clustering these simulations based on the dynasome variables that is form adjacency matrices to look at the dynamics fingerprint of those proteins. And then we are also considering devising methods for classifying the committee structure patterns that I've shown in the network. And the last step of this would be to construct a structure evolution space to complement the dynamic space. So the structure evolution space would somewhat mimic what I had shown in the second slide, a bipartite network of loops and domains that would have the dynamics layer on top. So now the last two are the caveats of our study that we need to address. So we need to sample more protein loops that should be representative of all the goal-level classes. So we have discovered 6 out of the 14 possible classes that have fold families, and then we should be striving for longer timescale simulations to capture the slower dynamics since they are related to function. With that, I'd like to thank the Blue Waters, the NCSA Blue Waters for providing us with generous allocations for 3 years rather. We also got allocation for a different project, and also the wonderful support staff, the technical team that helped us smooth issues and problems when we had them. And I'd also like to thank our collaborator, Dr. Falke Brader from the Heidelberg Institute for Theoretical Studies, and the Evolution Bioinformatics staff members. With that, I would like to take questions.
Interesting.